Welcome to NYU Langone Insights on Psychiatry, a clinician's guide to the latest psychiatric research. I'm Dr. Thea Gallagher. Each episode, I interview a leading psychiatric researcher about how their work translates into clinical practice. Today, I'm speaking with Dr. Mohammed Milad. Dr. Milad is a professor in the Department of Psychiatry at the NYU Grossman School of Medicine and director of the Behavioral Neuroscience Program. His research focuses on fear extinction, touching on everything from meditation to estrogen to transcranial magnetic stimulation. Clinicians treating patients with PTSD and anxiety disorders will find his work especially important and interesting. Dr. Milad, thank you so much for being with us today.
It's a pleasure. Thank you for having me.
We're just going to start by, we want to talk a little bit about what you're focusing on currently in your research.
So my research is focused on a very simple question. That is, how do we learn not to fear? That's just broadly speaking. And how does the brain form associations that we experience throughout our lifetime to enable us to function? And the key thing is about really fear and fear mechanisms and how the brain forms these associations across multiple brain areas. That's just generally speaking.
Gotcha. And why is fear such a problem? Or again, why are you trying to study it to understand it more?
Well, fear is everyone can relate to. Fear is a critical emotion that we all relate to, we all experience. It's just interesting to me from just a basic science point of view. You know, where and how in the brain fear is formed and fear is controlled and fear is regulated. So that just from a basic neuroscience point of view. And I was trained as a neuroscientist, and that's what I am, and that's what I am interested in. So that's just one angle. The other angle is obviously if fear goes unchecked, if we are unable to regulate it or control it, then it can lead to some difficulties for some of us to function, and that leads to psychopathology. So understanding the brain mechanisms of how fear is formed and interacts with other mechanisms or other systems like our cognition or memory or attention, and how when it goes unchecked, it can lead to disorders like PTSD, post-traumatic stress disorder, or anxiety disorders and things like that. So there are two, like I said, it's two angles. One is just because it's interesting, because I find it cool. And the other aspect is how it has a lot of implications to psychopathology.
Can you tell us a little bit about the studies that you're involved in that are ongoing currently and what you're looking at?
I always get myself into trouble with doing a lot of different studies. So we have a lot of ongoing projects here at NYU, all really focused on establishing circuits that learn to fear in the brain and how they are contributing or helping us understand psychopathology. So one project is looking at how the use of estrogen might help women with PTSD as an adjunct to exposure therapy, and we can go more into details about that. But basically trying to find ways in which we can improve clinical outcomes for PTSD treatment, especially in women diagnosed with PTSD. There's another project that we're doing is involving transcranial magnetic stimulation, and we're trying to see how we can use external devices like TMS to manipulate circuits in the brain with the objective of trying to enhance the capacity to control or regulate fear in humans, and how that might have future implications to improving treatment outcomes for PTSD. There are a number of other projects that are using some sort of modulation or another, neuromodulation or another, to study the change in our brain circuits before and after a given treatment in different populations. So I mentioned PTSD and using estrogen, the TMS. There's another project in which there's in collaboration with Dr. Naomi Simon at NYU Psychiatry, in which we're studying patients with anxiety disorders, and we're using meditation and studying the impact of meditation on the brain circuits in patients with anxiety disorders. So we look at their brains before they start the 8-week meditation course. We have them go through the meditation, and then we look at their brain after. So we're always looking at before and after changes in the brain circuits, as in the case of estrogen, for example. So we're scanning people before, we're scanning people after. With TMS, we're doing the same thing. And this is another project we're looking at using meditation to see if we can manipulate brain circuits. There's another project that we're trying to see the intersect between avoidance behavior, which we all know about and learn and experience and go through in life, fear, and reward, how the three intersect in the brain. So an example is if you go through a traumatic experience in a car, you have a serious car accident that, you got severely injured or hurt, you're going to, after that, try to avoid driving your car. But then, that avoidance subsides after some time, say a week or 2 weeks or a month, because you have to, say, drive to go to work. And so there's this thing of competition between, going over or getting rid of your avoidance response and your fear because of a goal-driven or goal-directed behavior that we need to do, which is, maintain our jobs, for example. So those are the things we're trying to understand in the brain, and we study this in using functional MRI to basically look at brain circuits. We develop paradigms that we have people go through in the scanner, and we study that dynamic interaction between these three. Let's see, other projects, and also OCD, obsessive compulsive disorder, and this is in collaboration with UPenn and Columbia, also looking at the impact of estrogens on exposure and response prevention in OCD patients. By the way, the estrogen project, I forgot to mention that it's also in collaboration with UPenn as well.
Can you tell us a little bit about, I think, so this podcast is really directed to psychiatrists and mental health clinicians. Mm-hmm. And I'm sure people will be interested to know, some of, us out here kind of doing the clinical treatments. Can you tell us a little bit about this relationship between estrogen and fear and what you know about it already and what you're trying to hopefully figure out in the long run? And then we can talk about implications for even clinical practice.
The study of sex differences have not really been carefully done for many years in the past. When we would basically, in animal models, we would just, the majority of studies used to look at male rats only. Female rats were always ignored or just not included because it's just easier to reduce variables. And because of the estrous cycle in the female rat, there's a lot of fluctuations up and down of hormonal changes. So scientists didn't want that, so they left the female rat out. And in human studies, people would just recruit sample of men and women and would put them together and average them and just look at their data that way, averaged, but without necessarily focusing on how men and women may differ. And so 20 years ago, I started doing this in human side because my PhD was in the animal models, and I only studied male rats. I'm guilty of the same, but I'm going to blame it on my mentor. So when I started to study humans, because I'm interested in the human brain and human behavior and the biology if you're from the human side, my first experiment, I noticed there were some differences between men and women and how they regulate their fear. So I do Pavlovian conditioning in my lab. That's what I study. That's what I do. I show people pictures of blue light, and I deliver a mild electric stimulus, call it a shock. And they learn that every time they see a blue light, they get shock to their fingers and they start not liking blue. And then I take away the shock and I just show them blue without shock. And then they extinguish that fear. Then I send them home, and then the next day they come back and I test their memory. So there's a fear associative state that we study. We call it conditioning. There's the extinction, and there's the extinction memory the day after. And we do all of this inside the MRI scanner, the fMRI scanner, and we're looking at their brain, and we see what lights up and how these things form and how these circuits are changing throughout these different phases of learning. And so what we found early on is that we noticed differences in how men and women form these associations, or how they express them, rather. And so I was intrigued, and I went to my mentor, Scott Rausch, at the time, and I said, Scott, there's, something is interesting here. Men and women are not necessarily expressing the same conditioned responses. They're not extinguishing similarly, there may be something to this. And I looked in the literature and there was really not much done. So we started looking at it. And what we found is that if we just study a group of men and a group of women and have them go through this conditioning and extinction training paradigm, and they learn to condition to form this association between the blue light and the shock just as fine, equal. And they also extinguish their fear similarly. And they also remember that extinction the day after. Okay. But when we look at women and we evaluate the women by their hormonal state, whether they went through this experience of condition and extinction in a high estrogen state naturally or low estrogen state. So they're near ovulation, for example, right? Or early in the cycle, in the menstrual cycle. We found differences within women. And then sex differences emerged so that women who ran through this paradigm, they came to the lab just when they're high estrogen, near ovulation, for example, or in the luteal phase, they were able to hold on to that extinction memory better, so that the day after, when they came back, their fear and anxiety, as measured by our skin conductance and some brain circuits, had higher activation, okay, compared to the women that went through this training paradigm in a low estrogen state. So that gave us an initial hint that there may be something related to estrogens or sex hormones, broadly speaking, maybe progesterone, but we're focused on estrogen and how that might impact the brain circuits that form these memories and store them. And so we basically done a lot of work the past 15 years, the summary of which is if we manipulate these hormones in women, if we give them estrogen exogenously, if we just give women a pill of estrogen and have them go through this extinction training, we can actually enhance their capacity to store that extinction memory better, which translates to having them better regulate and better regulate their emotional response to these conditioned stimuli and better able to express that safety memory, if you will, the day after. And so that's what's led us to this clinical trial that we're currently running, in which we recruit women with PTSD, diagnosed with PTSD, and then we scan their brains and we have them go through this conditioned extinction paradigm. And then when they come into the clinic, they go see the therapist and they go through prolonged exposure therapy. We give them a pill it's a double-blind placebo-controlled trial, either estrogen or a control pill, right before they come in to see the therapist. So basically the idea is to try to see if we can elevate estrogen during exposure therapy. Does that make the therapeutic outcome better? Are they able to go through the therapy session and learn from it more when we're elevating their estrogen? And instead of doing the 8 to 12 therapy sessions, we're reducing it to 6 therapy sessions. So that can we, instead of having the clinical trials go for 12 weeks, we're now doing it for 3 weeks, twice a week, and then see with or without estrogen. And then we're scanning after, and we look at the brain circuits and whether or not that changed because of this estrogen versus a placebo. So that's what we're doing, and we're doing the same clinical trial. We're not really, we're studying mechanisms of again the similar patterns and how estrogen might impact therapeutic outcomes in patients with OCD. Then again, that's a collaboration with Blair Simpson. She's the PI, along with Edna Fo as well as the PI. So we're a sub here. We're collecting the data from them, and we're going to be analyzing it. But that's broadly the objective here.
Yeah. And say, your hypothesis and things that you expect do happen. What do you think will be the clinical implications for psychiatrists or mental health clinicians?
They're twofold, in my view. One is having the patients understand and learn that the brain is plastic, and that when you're going through a therapeutic intervention, it's actually impacting your brain. So I think that's really powerful for a patient If I think I have an infection and I go and I get a blood test, right, and then I see my white blood cell count is high, and then you are my clinician and you give me this antibiotic and I take that antibiotic, and after a week or 10 days, I go back and I do a blood test. I feel better, but I'm not so sure, right? And if I then do a blood test and I convince myself by seeing that my white blood cell count is down, then I feel assured or reassured. So I think the patient's awareness and understanding that the brain is plastic and that therapeutic interventions don't just, they don't have an impact on your elbow. How psychotherapy impact you? How does it change you? It impacts your brain. So that's one thing. What I hope that we show is that various different therapeutic interventions actually do change the brain. And we're looking for is to show how and where in the brain these things happen. That's one. The second is that we're trying different interventions, whether it's, device-based neuromodulation or, different forms of psychotherapies or behavioral therapies or meditation or pharmacological agents and things like that. And again, the objective is to see how they impact the brain, but how we maybe are different. So maybe meditation works for you, but not for me. Whereas device-based neuromodulation is better for me and not for you. And so second objective is, as we move forward in this field, is trying to understand which therapies work and which therapies work best for you and not for me and vice versa. So try to kind of develop ways to tailor treatments based on who's it going to work for best versus not. And the third is the obvious, is that we're trying to develop novel interventions to try to improve or accelerate or current interventions. PE works, prolonged exposure therapy works, but not works for everyone. Or there's some, stress-induced reinstatement of traumatic experiences, for example, in PTSD. Well, if we can find ways in which we can improve on these current treatments, make them more, solid, if you will, if we can try to find ways to enhance the impact of these therapies by adding a pharmacological agent that can sort of cement that therapy better, if you will, in your brain and change neural mechanisms and enhance neuroplasticity in your brain, then that would be a very nice thing to show. And that's what we're really after. And obviously, device-based, like I said, trying to develop novel interventions and novel approaches for treatment.
Mm-hmm. Yeah, and I think, one of the themes that we've already seen in this podcast is this concept of personalized medicine, because we know things like PE work and we know that meditation works, but we're trying to now, it seems like, get more granular and how can we make it even better and how can we tailor it to the person and understanding what works for which person at the best rate, dose, time, or with an adjunctive. And I think that's one of the really powerful takeaways of how the field is moving. Would you agree?
Absolutely. And again, for me, showing that these therapeutic interventions, that they're skeptics, right? And about whether or not they work or how they work or where they, and I think if we show from a basic neuroscience point of view that they actually do impact your brain, there are synapses that change when you meditate. Now, how cool is that? The brain, parts of your brain actually, the structural changes occur when you do these things. I think that's an amazing achievement that we would have an obligation almost to report and show to the public and the patients that, look, this is what's happening to you and this is why you should stick to it and this is why it's going to be good for you if you do it, because it actually is changing your brain. Not just that it works, but how it works. Exactly right.
Yeah. As an exposure therapist myself, I get to experience the changes. But I think it would really bolster the treatment to explain to patients and to the world and to other, clinicians, this is exactly how it's working and the real changes that are happening in the brain.
Exactly. And you're again, with just other analogies of heart disease, and where your ejection fraction is at the 20%, and then you go through a triple bypass, and then your ejection fraction goes up to 45%. You have numbers, you have things to look at. Makes it tangible to the patient. And it's not just about your breathing is better and your life is better, but you actually can see it. So I think if we can, again, just to
Find ways to show it to the patient
find
too?
ways to show it to the patient. I think that's something that we're missing, and to show your patient, look, take a picture of their brains before therapy, go through the therapy, and then show them after. Wouldn't that be cool?
Yeah, more objective data, right? Yeah, and Dr. Marmor and I were talking about that. Like if you are diagnosed with cancer, you're not going to kind of get a one-size-fits-all approach, and you're going to want to see results and levels. And I think that likely will bolster someone's confidence even in their improvements and being able to, again, see measurable results that aren't just based on their self-report.
Absolutely, and it would give you guys, the psychiatrist and the psychologist, an organ. Because that psychiatry is the only branch of medicine without an organ, right? It's where are emotions coming from?
Right.
It's
time for our objective data.
Neurology has to share.
Yeah, right.
Yes.
No. So just to go a little bit into meditation, and I don't know if you have this data yet, but do you have a sense of how meditation is working to kind of change the brain?
Yeah, so this is the current trial that we're running with Dr. Simon is obviously ongoing, and I don't have any data to speak to that, but we have been doing some prior work with a collaborator at MGH, Sarah Lazar, and others, and there are it's a big field in neuroscience. We're looking at how meditation might impact the brain. And it does so in a number of different ways. Some of these data I'm talking about have been published, and some of it by us and some of it by others. Richie Davis is another name in Wisconsin, Madison, Wisconsin. That basically, the way we have data showing the way it works is that it causes or induces structural changes in your brain. It makes some parts of your brain bigger, thicker, which is sort of, crazy to think that it's possible, but it does happen. And particularly brain areas like the hippocampus and some areas in the parietal lobe that change. And those areas are important in memory and important in attention and also some sensory brain areas. So there's a change in the structure that is associated with meditations or people that meditate. And also change in function and functional connectivity, basically how brain areas are wiring together. And people who meditate tend to have sort of better communication between nodes in the brain that are important in attention and in sensory processing and in memory. What that suggests is that what meditation is supposed to do is makes you aware of the present moment, of yourself, of your surroundings, and the data so far suggests that there is support for that it changes brain areas both structurally and functionally so that they are better able to communicate with one another.
And so in doing so, the goal is that, do you need to practice this kind of for ongoing in order to have these changes be long lasting, or do you think that the changes might happen and then it carries with them for the long term?
Well, yeah, practice is essential. It's sort of like you're practicing to be a better basketball player or a football player, or you need to practice. So the way the brain works is, it causes the plasticity that have to happen, and there are changes in connections, and the structural changes and the functional changes don't just happen overnight. So it just takes some time for the brain to change those things, and not only change, but to make those changes permanent. And the way that happens is you have to basically go through that meditation practice and the homeworks and so on for that to, again, to be maintained in the brain. And that then you were able to utilize that not just in your meditation, but across different modalities of your life, different aspects of your life.
Mm-hmm. Yeah, and I think it's something that many therapists would suggest to patients, but again, having some more measurable data on how it works and that there are these changes that happen, again, can bolster the treatment moving forward.
Absolutely. Yeah, and this idea that, again, I hear it a lot from when I talk to people that, there's something about neuroscience, but then there's something about psychology that they're different. And I always get confused by that because I don't know psychology. Where's psychology? it has to be coming from an action potential. Otherwise, I don't know where would that be coming from.
And so for people that are listening, psychiatrists and psychologists, therapists who are listening, from your work, what is a takeaway that you would like people to focus on in, kind of in their clinical work from everything? I don't know, from these studies that you're working on, but maybe studies in the past or things that you know about the brain. It sounds like helping people to realize, okay, the brain's plastic, real changes can be made. Anything else that you think, again, someone can take with them today and have incorporated in their clinical work?
I think, at least the way I see it here and how psychiatry is being practiced at NYU and how it's being taught and the residency programs, is that I feel that clinicians are becoming more and more aware of what neuroscience is doing, and there's more dialogue between psychiatry and neuroscience and psychology and neuroscience. And I think that dialogue is very important in learning about each other's language, if you will. And I'm better understanding, for example, what is PTSD and what is anxiety, because I was trained as a neuroscientist. I didn't learn about any of these things. And psychiatry and psychology in the past, I get from my understanding, had minimal training in neuroscience and neuromodulations and things like that. And I think that takeaway for my fellow clinicians, my wife is a psychiatrist, and so we talk a lot about these things, is that when we talk, we better understand each other. And I think that better understanding between the two is going to ultimately help the patient, because your improved understanding of not just your field and your training as a clinician, whether you're a psychologist or a psychiatrist, and all of the inpatient and outpatient amazing work that is done and that you acquire. But I think some better understanding of neuroscience can also help your patient, in that you can there's more of this psychoeducation to teach and tell your patients that what you're doing isn't just talk therapy. It's talk therapy that impacts their brain. And you can say that, and you can say that with confidence, you can say that with knowledge, you can say that with a base, a neuroscience base. And these are the brain areas, and that's how they talk, and that's how, when we do this therapy over time, this is what it's going to do to your brain. And so I think that's something that I see done here, and I think there's more to be done as well, to just help us all better educate one another and communicate with each other, so that we can ultimately impact the patient and help the patient.
Yeah, and kind of more synergy there, and it sounds more of a both and approach rather than an either or, being less siloed and more collaborative in how we understand and how we approach.
Absolutely.
Yeah. And I did want to go into, just briefly, I guess, can you explain to us a little bit about TMS and your work with TMS and fear and what you're understanding there?
Yeah, so I mentioned action potentials of how just the brain is just a bunch of neurons that talk to each other. And so more than 20 years ago now, I did this experiments with rats in which I recorded from a brain area, prefrontal region, when the rat is learning not to fear, and found this increase in brain activation. And the higher the increase in this activation in this brain area, the less fear that animal expressed, or less threat responses that animal expressed. We got excited about that. And my mentor, Greg Quirk, said, what if we try to manipulate the behavior of the animal by putting electrodes deep in that same area and deliver artificial stimulation to that brain region? Would the animal's behavior change? And I said, okay, let's try it. So we did that, and we artificially implanted a safety memory in the animal's brain. And we conditioned the animals. The animals were afraid. They were showing threat responses, conditioned threat responses. Then we delivered these microstimulation, very, very brief stimulations. Artificially, I turned the switch on, put the electrodes in there, turned the switch on, and the animal wasn't showing fear, wasn't showing these threat responses. So we got really excited with the idea that if you do some stimulation in the brain, if you artificially manipulate neural activation, you can actually impact behavior. Now, that's a crazy thing that some people might not like or might not feel comfortable with, but that's the reality, that if you put devices on someone's head or if you put deep brain stimulation in patients, and that's already been done, and a lot of work has been done on this, DBS and so on, you can change human behavior. And so our objective with the TMS, and I don't want to go into that territory, but our objective with the TMS is, if we identify the brain areas and the brain circuits that are critical to inhibit fear, can we put an electrode on the surface? Can we put this coil, a TMS coil, on the surface and turn the stimulator on when you see that blue light, for example, that I told you about earlier, that I conditioned you? You see blue light and you're like, uh-oh, shock is coming. Well, can I turn the stimulator on and hit a specific target at a specific time, target in your brain? And would I be able to manipulate or change your expression, your conditioned responses to that? And we actually did that study a few years ago and showed that in a small sample of participants that is in fact possible. And so now we're trying to do this at a much larger scale, and we're trying to fine-tune the stimulation parameters and the location of the stimulation across different targets in the brain to see if we can, this is from a mechanistic point of view, to see if we can modulate brain responses to stimuli that have been conditioned with exogenous or, with the TMS, and how we can manipulate not only the circuits and how they interact, but how we can potentially manipulate behavioral expressions or psychophysiological expressions of those conditioned responses in humans. And the objective is that we would use this as a form of therapeutic intervention for example, PTSD patients.
Mm-hmm. And it seems like the timing and the specificity is really important. So as you look down the pipeline, how, getting this research into, the hands of people who could do something about it, we know that there's a pretty big lag between, research to practice. It sounds like there should be, though, some collaboration across providers, because there is, it seems like there is some specificity to what you're doing, both with timing and kind of focus.
Yes. So, for example, if you think about, absolutely right, because, TMS is used for treatment of depression. It's FDA approved, and it's also now being used for PTSD treatment. But the way it's done is you put the coil at some particular location in the left prefrontal cortex, and you turn the stimulator on, and for a certain amount of time, and you do that repeatedly for X number of days, and then that's it. It's done. And it's effective. It's useful, and a lot of patients get better, which is wonderful. But can we do better? And instead of having it on for a long time, and like with PE, can we reduce the sessions? And can we make it very, very specific? Can we help people with specific phobias, for example, spiders or snakes or heights or things like that, so that we don't just turn on the stimulator while they're just looking at the white wall or thinking about their grandmother. It is basically you show them the stimuli that are causing the discomfort, the anxiety, and things like that, and you deliver the stimulation at that time. And so that you make it more precise, you make it mechanistic, and it's targeted. And also better location of where we're going to be delivering those stimulations, and potentially also maybe reducing any unwanted effects or side effects and things like that. That's the idea. That's the future, is to try to have these TMS sessions be more precise timing-wise and also location-wise.
Mm-hmm. Yeah, and it sounds like, again, getting into that personalized medicine approach, which will be better for both clinicians and patients. And the last kind of area I want to get into a little bit is, how do these findings and the things that you're researching, how do they fit together, or maybe they don't yet, with research into psychedelic-assisted psychotherapy for PTSD?
I don't know the answer to that yet. I'm not sure that there is an intersect at this point. I think research in the psychedelic domain is just beginning. I think that in the end, what we will probably find is that it's just a different venue to impact the same circuits. So, whether it's meditation or whether it's TMS or whether it's estrogen or psychedelics, we have one set of brain areas, right? We're not gonna, discover a new brain region behind your left ear. We have what we have. So I think it's just a different way. I think it's going to be maybe different mechanisms that psychedelics are impacting or having their impact. There could be a broader impact at different networks. I'm not so sure. I don't know. The answer is I don't know. But I think it's probably going to, what we'll end up finding is it's a different way to modulate the same or similar circuits, but with different mechanisms.
Yeah. And I think in some ways it's good that there, like you said, there is only, the brain has boundaries. So it's not endless in that part of the discovery. But I guess when you're thinking about, in your career, is your $1 million question kind of always like, why do we fear? And is that what you kind of are continuing to try to understand?
No, I like fear. I don't know if I mentioned this at the beginning or not.
No.
I think fear is good. I mean, it's too much fear. It's the continuum. Yeah, I think of it as in a spectrum of like colors of lights. I think fear is okay, and fear is useful for us. If you're not afraid you're crossing the street in Park Avenue, you'll die or you'll get hurt. I think fear is important. I think that too much fear or unable to regulate it or contextualize it, there's no reason to fear a snake when it's behind a thick glass in the Bronx Zoo, but you better run if you're hiking in Colorado and or somewhere and you see a snake coming at you. You go. No, I don't want to be afraid of snake. No, you'll die. So I think maybe the $1 million question for me is, why do we differ in how we regulate fear? I'm interested in individual differences and our abilities to control fear and what happens when we're unable to regulate it. That's what I'm interested in. That's what I'm continuously curious about.
Yeah. And if, I don't know if you have any suggestions, but for, again, many of us as clinicians, utilize meditation in the work that we're doing and personally. Are there certain resources for meditation that you like over others or that you found in your research are more effective than others?
So I don't meditate. I can't meditate. I tried. I fly. So I think that the key thing is to engage your sensory processes in one way or another. I think one of the aspects of meditation is just refocusing and reshifting your attention. And so in terms of resources, I don't know the resources because I don't meditate. But when I get tense or anxious, I fly for fun. And actually going to fly makes me relax. And that may be crazy to say or hear or people understand that, but to me it just disconnects me from everything else. When I'm up in the plane and I'm talking to tower and I'm controlling the yoke and the power and writing down and changing frequencies on the radio, if I'm anxious about anything outside or if anything is bothering me, I don't think about that because I can't, because I'm forced to focus on the present moment. The present moment is flying the plane. And to me, that's meditation. It's just a different form. Yeah.
Yeah, but it's a different form, and yet it sounds like some of the core ingredients of being able to be fully present in one place and focused on, again, what is happening in the here and now, which could be, maybe some interesting future research and understanding what is a meditative space for an individual.
Yeah, I really think there's a key element of an interrupt signal, if you will, that if you're continuously perseverating onto your thoughts of anxiety and fear and so on, if there's a way in which you're able to interrupt that, and meditation is a form, and for me, flying is my form. And then it breaks that cycle. And then when, if I'm really upset or something, I just go fly. And when I come down, I just feel really exhausted. And it's almost like going through that state of meditation. And I think maybe that's another aspect that we haven't really paying attention to, is all these different ways, whether it's therapeutics or things like that, is just trying to give the patient a power to find a way to put a stop sign, and then say, okay, anxious feelings and, perseverative thoughts, stop. And if there's a way you're able to do that, then that gives you an ability to control. And if you're able to control, then some anxiety and fear and so on will subside, in my opinion.
Yeah.
I'm not a clinician, so just make sure I'm a basic neuroscientist.
Yeah, but it's like, again, I think we're coming back to this point of it's not a one-size-fits-all. I did a media story recently on how they found a correlation between, managing anxiety and bird watching. And I bet that's not for everyone. But as we're talking about it, kind of being focused, being meditative in the sense that it brings you to the moment, you're able to interrupt that cycle and be engaged with what's in front of you. Like you said, helping patients maybe to figure out what that is for them, from trial and error, and then ultimately from, further study of what's happening in the brain will be really powerful for our field moving forward.
Absolutely.
Well, thank you so much for being with us, Dr. Milad, and we'll definitely have you on in the future.
Look forward to it. Thank you.
Thanks so much again, Dr. Milad, for that conversation. If you enjoyed this episode, be sure to rate and subscribe to NYU Langone Insights on Psychiatry on your podcast app. On the next episode, I'll be speaking with Dr. Naomi Simon about her research into using yoga and mindfulness to address anxiety disorders. For the Department of Psychiatry at NYU Langone, I'm Dr. Thea Gallagher. See you next time.